For thousands of years, these symbols covered the walls of ancient temples and forgotten tombs, hieroglyphs. But to humanity, they were silent. But the greatest code humanity ever deciphered wasn't written in stone. This code exists inside every living cell, buried within a molecule called DNA, a language older than humanity itself. And to truly understand it, it's time to go down the rabbit hole. From microscopic bacteria to blue whales the size of buildings, from forests to humans, all life is constructed from the same hidden algorithm, an internal blueprint. And that blueprint is encoded in DNA inside every cell in your body. To understand this code, we first need to look at the structure that carries it. DNA, a twisted ladder unlike anything else in nature. Two strands, spiraling endlessly around one another. Like a spiral staircase, a shape so strange and so precise that it became the symbol of life itself. Hidden within the spiral are four molecular structures known as bases: adenine, thymine, cytosine, and guanine. Four chemical letters, usually reduced to something much simpler: A, T, C, and G. Just four symbols. And yet, from them, nature writes every living thing that has ever existed. They are the code of life. But there's a rule hidden inside this code. Adenine always binds to thymine. Cytosine always binds to guanine. Like matching symbols in an ancient cipher, each letter can pair with only one partner. This hidden symmetry is what allows the code to be copied, because DNA itself rarely leaves the nucleus. The original blueprint is too valuable, so the cell creates a temporary transcript instead. The double helix begins to unzip, and a molecular machine moves along the exposed strand, reading the genetic sequence one letter at a time. From this process, a new molecule emerges: RNA. Unlike DNA, which exists as a double strand, RNA carries the message in a single line of code, a temporary copy of the original instructions, and the copy follows the same hidden rules embedded in the DNA itself. Each letter pairs with its matching counterpart, allowing the genetic sequence to be rewritten, one symbol at a time. This entire process takes place inside the cell's nucleus, a process known as transcription, but the message is not meant to remain there. The RNA leaves the nucleus, carrying the instructions into the cytoplasm, where the next stage begins: translation. But before we can understand that process, we need to understand the molecules that make life possible: proteins. Proteins form much of the structure of living organisms. Your muscles, your skin, even traits like the color of your eyes. And every protein is built from smaller components, molecules known as amino acids. There are many different kinds of amino acids, each with its own shape, its own chemical properties, and its own role in the architecture of life. Every protein is defined by a specific sequence of amino acids, one added after another, in a precise order. Change the sequence, and you change the protein itself. But this raises a question. How does the cell know which amino acid comes next? How does it know the exact order needed to build a muscle, a neuron, or a molecule inside your eye? The answer lies in the genetic code, hidden within the DNA all along. The RNA molecule created inside the nucleus is known as messenger RNA, a fitting name. because its job is to carry the instructions copied from DNA. Once it reaches the cytoplasm, it encounters one of the most remarkable molecular machines in nature, the ribosome. And this is where the genetic code begins to reveal its true purpose, because the same pairing rules that allowed the message to be copied are about to be used once again, this time by another molecule, transfer RNA. Inside the ribosome, the message begins to be read, not one letter at a time, but in groups of 3. These 3-letter sequences are known as codons, and each codon carries a specific instruction. To the cell, they function almost like words in a language. But reading the message requires another player, transfer RNA, a molecule capable of recognizing these 3-letter patterns. Using the same pairing rules that govern DNA itself, transfer RNA binds to the messenger RNA with remarkable precision. If the message contains the sequence AUG, only its matching counterpart can lock into place, like a key fitting a specific lock. This matching sequence is known as an anticodon. But transfer RNA does more than recognize the code. Each one also carries a specific amino acid. In other words, every time a codon is matched, the correct amino acid is delivered to the growing protein. One codon, one instruction, one amino acid added to the chain. And as this process repeats over and over again, the protein slowly begins to take shape. This entire process is known as gene expression. The journey from information to matter. It begins with transcription, where the instructions stored in DNA are copied into RNA, and it ends with translation, where those instructions are finally converted into proteins, a molecular language, first copied, then interpreted, then transformed into the building blocks of life. The same genetic code is used by bacteria, trees, whales, and humans. But perhaps the most extraordinary thing about this language is that it is nearly universal. The same genetic code is used by bacteria, trees, whales, and humans. Life, in all its astonishing diversity, writes itself using the same molecular alphabet. And that raises a profound question. Why would every living thing on Earth share the same language? The most likely answer is that they inherited it from a single ancestor that lived billions of years ago, a distant organism known as LUCA, the last universal common ancestor. Every forest, every ocean, every animal that has ever walked, swum, or flown, all connected by a code that has endured for billions of years.